Hey Team Bio, we are deep into our study of DNA, its form, and its function. In class over the next few days, you are going to be figuring out how DNA is replicated, so how we go from one double strand of DNA to two double strands of DNA and keep the same base pair sequence. This screencast is all about the central dogma of DNA, or of living things, the central dogma of biology, I guess I would say, which we know that DNA encodes the traits and behaviors that we transcribe into RNA and then translate into proteins, which actually create the characteristics of living things. This screencast is going to be broken into two pieces. The first episode, if you will, is going to be transcription, where we talk about this DNA to RNA process. And the second part is going to be translation, how we read that RNA and we create a protein. I have in here the secret of life in parentheses, because really understanding the process of transcription and translation allows us to hack living systems, to create the DNA that we want, to change the proteins that we want to change, or move DNA from one living system to another and get, so for example, jellyfish produce a protein that is fluorescent, green fluorescent protein, GFP. We can take the DNA for GFP, stick it into another organism, and start making other organisms glow. So understanding this process is really super powerful understanding, and it's also, as they say in Spider-Man, with great power comes great responsibility. This has profound implications for the world and the direction that the world is going, with the advent of the ability to hack the genome. Okay, so let's get right to it. Transcription. Oops. I'm gonna make this a little bit bigger. Sorry, Campbell, this is tiny. Okay, transcription is the process of taking DNA, reading it, and synthesizing from it RNA. Okay, you all know that DNA is a double-stranded molecule that every side is composed of a sugar-phosphate backbone and then nitrogenous bases. So sugar-phosphate backbone, nitrogenous bases, sugar-phosphate backbone, nitrogenous bases. And there's a specific base pairing. A always binds to T, and I'm actually going to do 2 there because there are 2 hydrogen bonds between A and T, and G always binds to C. So if we were to pull apart this strand of DNA, if this is an A nucleotide, this would be a T nucleotide. If this is a T nucleotide, this would be an A nucleotide. If this is a G, then this is a C. If this is a C, then this is a G, so on and so forth. Okay, so we have this specific base pairing, and there's also base pairing between DNA and RNA. In DNA, we have the nucleotides A, T, G, and C. And in RNA, we have a different nucleotide for T. It's called U, uracil. So if you have an A in DNA, it's going to bind to uracil in RNA. T will bind to A in RNA, G will bind to C in RNA, C will bind to G in RNA. Okay, so not only is there base pairing between DNA nucleotides, but we can also have the same interactions between nucleotides of RNA. So what happens here? I think you can kind of guess. What's going on here is we have an enzyme. It is called RNA polymerase. RNA polymerase hops on to the DNA in a specific place that indicates the start of a gene. And it starts to synthesize a immature messenger RNA. The M here stands for messenger. And this is immature right now. We are going to mature it in a bit. So the way that this mRNA is synthesized, now it kind of looks like this is just a ribbon, right? But if you actually look over here, you can see that there are teeth in this ribbon, and the teeth are all nucleotides. Let's just give them some random names. Or not random, but say this is C, this also looks like C, this looks like G, this is an A, and this is a T. So you can see here that we have, I've just picked out names for these guys, and if we were to look at the DNA, we would see that this would be a G here, whoops, this is a G, this is a G, this is a C, this is a T, oh, wait, this is wrong. This would be a U, sorry, mistake. So this would be an A. Okay, so the DNA is a series of nucleotides, and in order to synthesize an RNA, you find the complementary nucleotide and you add it to the 3' end of this strip. So the next nucleotide, again, looks like it's a C in the DNA. So there's going to be some RNA nucleotides floating about in the cytoplasm, in the nucleus. And so the next nucleotide that's added to the sequence here is a G. And then next up, looks like this is an A. So we are going to add a U here. So I hope you can see that this is a specific sequence of RNA nucleotides that's built off of a specific sequence of DNA nucleotides. And RNA polymerase is going to keep on zipping along the DNA in this direction. As it zips along the DNA, it's going to open up the helix, and we're going to continue to transcribe the DNA and from it make a sequence of RNAs that is very specific. Okay, so ultimately, RNA polymerase is going to come to the end of the gene. And when it does that, it is going to fall off the DNA, and it is going to synthesize what we call a poly-A tail. So it's just a bunch of adenines, or the nucleotide A, in a row. So we call this a poly-A tail. And also, kind of not pictured here, is another sequence is added to the very top of the mRNA, and it is called an MG cap, a methylguanine cap. So it's a modified G nucleotide. Okay, so adding the methylguanine cap and the poly-A tail are part of how we go from immature to mature. But there's one other important piece that has to be accomplished before this mRNA is ready to leave the nucleus. And that is a process called splicing. So, splicing is when we take an mRNA message and we cut it up. There are some pieces that are cut out of the message. They're called introns. They're cut out of the mRNA and left in the nucleus. So it's kind of confusing that they're called introns, but it's left because they're left. The sequences that are cut out are left in the nucleus. Exons are pasted together when the introns are removed and exit the nucleus. So, if we were to look at this mRNA, you can see that everything that's in dark red is an exon. So, these components are going to be glued together, and they're going to be part of our mature mRNA. Introns are cut out, and they're shown here in light pink. And they're left in the nucleus, and they're going to be degraded, just broken back down into free nucleotides here. Okay, so why is this process important? Like, why even do this splicing thing? Why cut out sequences? Why not just have the DNA that has the proper sequence without cutting anything out? And part of the reason for having splicing is because it allows one sequence of DNA to be modified in a couple different ways to create a couple different mRNAs. So splicing allows for one DNA sequence to lead to many mRNAs. Depending on how this is spliced, the exons and introns might change. So in this mature mRNA, everything in dark red is going to be part of the message, and everything in light pink is going to be cut out. But in another splicing of this same sequence, we could take out this portion of the message and leave in this portion, and that would change the ultimate message that it is sending. So splicing allows us to take one DNA sequence and turn it into multiple mRNAs depending on how it is processed. Okay, so ultimately what we have here is a mature mRNA. This is mature, and it's leaving the nucleus through the nuclear pore. So it's got the methylguanine cap on this side, it's got the poly-A tail on this side, and it's composed of all exons. The introns have been removed and they remain in the nucleus. Okay, and that takes us to the end of transcription. We have read DNA and we have sequenced an RNA message that we are exporting from the nucleus. And next screencast is going to be about how that mRNA is read and a protein is synthesized from the information contained within the message. Okay, that's all.